bit for a little bit of contextual review before we start talking about immunopathology. So first of all, we had talked a little bit about opportunistic pathogens and the fact that they are defined by an organism that might not be a problem in a certain place if it's in its correct niche, but it could be a problem if it's in its outside of its niche, in an ectopic site. And we talked about a number of conditions or situations where that might be possible. So when we talked about what pathogenicity means as a term, this is sort of one of those yes or no things. You either are a pathogen or you aren't. But sometimes organisms that are kind of regarded or considered to be commensals, like Candida albicans that we talked about, like yeast, it can be considered, could be considered a commensal, or it could be a pathogen as well, right, depending on where it is in the body or what the host immune response is, or both. So Candida albicans, it colonizes what kind of sites? Give me some examples of sites where you can get Candida albicans colonizing. Yeah, in the mouth, low levels. GI tract, yep. Yeah, your genital tract. Yeah, so it's in all those places, and it's kept in check. And if you introduce dysbiosis, it can cause overgrowth of Candida, right? And you get problems like yeast infections in various places of the body. So it goes from being a commensal to a pathogen in a context-dependent way. Those were those examples we talked about where it's not just where the yeast is moving it from a site where it's kind of okay depending on host response to like the blood where it should not be. You can clear it if you have a good immune response. But also, even if it is where it is normally in its normal niche, like in the mouth, the host immune response itself can cause some issues, like a weak immune response, for example. So just wanted to kind of underscore those points, and it's a kind of a good segue to talking about immunopathology. We're talking about how important the host immune response is. Sometimes, in particular, when we're looking at organisms as we go through this course in Block 3, for example, and talk about different types of bacteria, some of them, they don't pack a lot of virulence factors, which we haven't talked about yet, but we will shortly. So these guys, for example, kind of on that left side of the spectrum, sort of considered to be harmless commensals for the most part. But sometimes just the fact that they're introduced into a site where they shouldn't be, it can elicit a strong immune response. So they can cause disease without having very many virulence factors themselves, just by virtue of triggering the innate immune system, essentially. So let's go through some of this stuff. We've already talked about some factors of immunopathology a little bit when we were talking about immunology in Block 1. And you'll certainly hit this again next semester in pathology, but yeah, let's just go through a couple of these. We've talked about fever a little bit. Most of these immunopathologies have some beneficial quality, even though they're associated with a lot of pain and suffering. With fever, we talked about this one. What is it supposed to do? Yeah, it's supposed to have a suppressive effect on those pathogens. So those cytokines, those pro-inflammatory cytokines get released during like a bacterial infection or a viral infection. They will transduce some signals that cause the hypothalamus, the part of your brain that controls and regulates temperature. It'll cause it to reset that thermostat to a higher temperature as a result. But then, of course, you have those problems associated with super high fevers in particular that get to be dangerous for these people with high fevers. What about malaise? What is malaise?
Fatigue, tired.
Tired, yeah.
Lethargic.
Lethargic is tired, yeah. What else is associated with malaise? It's not just tired. Like I was tired last night, but I didn't have malaise.
Sick, generally, don't feel good.
Yeah. So what could possibly be a good positive aspect of malaise?
It tells you something's wrong.
And it tells you not to. Yeah, it tells you to just recover. You need to rest and recover. That's important. Because when you are fighting a particularly serious infection, that's going to expend a lot of energy. Your body takes a lot of energy when you're fighting an infection. You feel that. So when your brain is sending you these sickness messages that you feel sick and you have malaise, it's telling you to stop. That's not a good time to exercise. It's probably not a good time to go to work. It's probably not. Just stay home and rest. so your immune system can have time. And that can take, some of you are smiling because you never get that time, do you? In the modern world, you're always like encouraged to push through it and go to work. But you shouldn't. You should stay home if you're sick. Tissue damage at the infection site. If you're talking about, let's say, a localized infection, like a skin and soft tissue infection in particular, some of these cast of characters we've talked about before, we have our immune cells and some complement protein system. Which of these cells do you kind of most associate with tissue damage?
Neutrophils.
Neutrophils, yeah. They cause direct damage, and they also cause indirect damage just by recruiting into that area and causing blood vessels and capillaries to expand as they come into that area. With complement, particularly some of these complement factors like C3 and C5 that we didn't talk about, but those serve as anaphylatoxins. They have specific receptors on innate cells like basophils and mast cells. So when those complement anaphylatoxins bind to mast cells and basophils, it causes release of histamine. And histamine is one of those very potent vasodilators. It has other inflammatory reactions as well. So that can cause damage. Very closely related to that next bullet about vascular permeability and potential for capillary breakage. When you have that heavy infiltration of immune system or immune cells into an infection site, over time, if you have enough, that's going to cause some pretty serious damage to those blood vessels. And that can cause those capillaries to start bursting in those areas. So a little bit of internal bleeding at those sites. But what was the purpose? What's the benefit when you have permeabilized vasculature and blood vessels? Yeah, diabetes is you got to get those immune cells into that site in order to clear the infection. So that's the trade-off. Most of this is just a double-edged sword. What about edema? What benefit? What is it, and what benefit does it confer? It is swelling. Yeah. How does that painful nuisance in the process help you clear an infection? A little bit more specific.
If something's painful, we don't use it.
Okay.
And it
Yeah.
improves blood, which is going to clear out the bad and bring in the good.
The increase of blood flow to that area is sort of related to vasodilation and the expansion of those blood vessels. But if you're just talking about leakage of fluid into interstitial spaces, it causes edema, whether it be in tissue or more importantly if it's happening in the lungs, then like what benefit would that serve? Taken up to the lymph nodes?
More
Potentially.
WBCs can get there.
So what can?
So more WBCs can get there, like white blood cells.
Yeah, that's pretty much what's happening. You're expanding some space in that area because, and that's why you can see visible swelling, right, in some of those areas. Or if you have lymphadenopathy, you can actually see those lymph nodes expanding out. But that is the increase or infiltration of immune cells into that space, and that fluid, that leakage of interstitial fluid into that place. That's mostly undesirable, but that's also accommodating, that swelling is also accommodating the influx of immune cells into that area. With pus, that's just the collateral damage on the battlefield. That's not doing anything good for you. What is pus?
Dead neutrophils.
Yeah, a lot of dead neutrophils. What else? The bacteria, yeah, it's bacterial infection. Anything else?
Damaged tissue.
Damaged tissue, yeah. Let's say you have a hangnail, just a tiny little infection, not a big deal, but you ever had one get infected, you can see that inflammation. That's going to be a lot of neutrophils, it's going to be the bacteria, and it's going to be some of that steroid tissue or damaged tissue at that site. Given that this is a temporary thing, you should be able to clear that pus out. Some purulent infections, purulent means pus generating, will last for a while, but pus is something that your body should reabsorb. Like if you have a zit, that's just a small amount of pus, and you should be able to reabsorb that. If it's a really bad purulent infection, like a carbuncle or furuncle or something like that, or boil, you might need to get that thing lanced and possibly treated with antibiotics. But a couple other mechanisms of immunopathology damage caused by the immune system. Granuloma formation. This is the body's way of cordoning off and dealing with infections that have a high likelihood of disseminating and spreading throughout the body. The classic example is Mycobacterium tuberculosis, where unfortunately this is an intracellular bacteria, it can infect macrophages, and even though it's associated with lung infections, it's a pulmonary infection, if it disseminates throughout the body, that can be a very rapidly fatal condition. So it's an old, ancient disease. We've been co-evolving with Mycobacterium tuberculosis for millennia, and this is what our body, our immune system has developed as a strategy. It's like, I want an analogy. You guys seen Outbreak? That movie with Dustin Hoffman, Rene Russo?
Nope, never seen it, I'm going to.
Oh, touche, man. Okay, fair. Okay, well, at least watch the synopsis on YouTube, like I personally doing. No, so watch Outbreak. Fun little more excitement. But plot spoiler: there's a big viral outbreak. It's some like scary Ebola type thing that's coming to the country, and there's a town that's like ground zero for this viral outbreak. This town is it's called Cedar Creek. And this evil general, because of course the military takes over, because they always do, right? This general's like, we gotta bomb it, we gotta firebomb this town. Right? You saw it, right, Thomas? You see it?
Maybe, yeah, that sounds familiar.
Okay. Anyway, everyone's arguing against it, like, don't blow the town, because there's guaranteed gonna be some infected people that escape this town, and then it will be for nothing. So they're like sending the order for this plane, this military Air Force pilot, to drop a bomb on civilians in this town, and Dustin Hoffman's character is trying to argue them out of it. The analogy to granuloma formation is this is the body's way of sort of firebombing a small section of the body in order to get rid of the pathogen, because it doesn't want it to disseminate and infect the rest of the country or the rest of the body. All right. The trouble is with granulomas, particularly with Mycobacterium, some of those cells do sometimes escape the granulomas, and then you can get reactivated tuberculosis in disseminated forms, and you can still die. Okay. But it's not just TB. You can form granulomas around other bacteria. Lymphogranulosum venereum is one. Some parasitic infections, schistosomiasis, leishmaniasis. Some fungal infections that affect the lungs. So this is a commonly used strategy for different difficult to handle pathogens. But in the process, all that tissue that's inside that granuloma is destroyed. It is liquefied. In the lungs, this can be a problem if you have multiple granulomas. You start losing lung capacity. So, some serious pathology related to granuloma formation.
Is it like, I guess I'm thinking like granules like being shot out. Is it kind of like almost like a glass dome, not glass, but a dome over the affected area that kind of keeps it contained and kind of kills it from the inside?
Yeah, it's a three-dimensional sphere granuloma. So you have a foci of bacteria or parasite or fungi, and then this is mediated by CD4 T cells, but it has a lot of big cast of characters. It recruits epithelial cells, other parenchymal cells in the area. So if we're talking about the lungs, there will be fibroblasts involved, there will be alveoli involved, there will be lots and lots of alveolar macrophages, CD4 T cells. And then they start organizing themselves in such a way where it just forms a barrier around those bacilli so they don't disseminate. But that is supposed to be a permanent solution. What happens eventually is sometimes your immune system starts waning or you get sick and your T cell response starts crapping out, and then you have a rupture of those granulomas. And if there's anything surviving in them, they can escape. Yeah. Cytokine storms, we've talked about that a little bit, but that's that out of control, inappropriate negative feedback loop that just escalates and becomes extremely dangerous. It can be caused by things like superantigens or the en masse release of endotoxins from gram-negative if you kill them rapidly. Autoimmune responses, I have two examples up here of autoimmune responses. We briefly talked about what these are, but these are both sequela of different bacterial infections. What does the word sequela mean? Yes, sir?
Secondary infection.
Is infection the right word? Yeah, a little bit broader. It's actually not due to an infectious issue anymore. It's a problem. It's a secondary problem that arises after infection. Two examples to illustrate what that might look like. One is rheumatic fever. We talked a little bit about people who get strep throat, right, which is an uncomfortable thing, it's a sore throat, but very low fatality rates. What happens is your body will raise antibodies, your humoral response, you will get antibodies against a particular protein that is associated with the causative bacteria, Streptococcus pyogenes. And when you raise these antibodies, sometimes a small number of people, this doesn't happen in everyone, but some of those people have antibodies against that protein that unfortunately are cross-reactive to heart tissue. So those antibodies, like weeks later, potentially months later, can start attacking your own heart tissue. And that condition is called rheumatic fever. That's one of the reasons why strep throat is more serious than if it were to be just a problem in and of itself. We also worry about the sequela that can be caused afterwards. So because of that, we have the central criteria and we treat it a little bit more seriously than we would otherwise. Another really common example is Guillain-Barré, which I mentioned also last block. But Guillain-Barré is the sequela of a bunch of different microbial infections. The biggest one is this enteric pathogen that you may have heard of called Campylobacter jejuni, where about 30% of people who have Campy infections, this intestinal bacterium, will go on and develop the sequela of Guillain-Barré. But other intestinal pathogens can do that as well, and some herpes viruses can elicit Guillain-Barré. Essentially what's happening here, though, is the same deal. You are raising antibodies inappropriately against some immunogen associated with a pathogen. In the case of Campylobacter jejuni, this is a gram-negative, and its cell wall has LOS. I'm sorry, I made a mistake there. I put lipopolysaccharides, but lipooligosaccharides are essentially the same thing as LPS, which we've heard about before, right? Just a shorter saccharide layer than LPS. So your antibodies will be raised. Some of them will recognize a host cell motif that's very similar to LOS. And this host cell motif is present on gangliosides. Those are nerve cells, so your antibodies will start inappropriately responding and reacting to nerve cells, and that causes ultimately the demyelination of those nerve cells, which can interfere with signal transduction, which can in turn lead to paralysis and loss of sensation. So Guillain-Barré is usually considered to be much more serious than the initial infection that caused it, but that's another autoimmune downstream sequela. All right, so that's immunopathology. We've talked about endogenous infections, opportunistic infections. Next, we just want to sort of transition over to talking about portals of entry for exogenous or mostly exogenous infections or bacteria that can cause infections from the outside. So with most of the examples, it's pretty intuitive that we're talking about ways of these bacteria getting into the body, these different portals of entry. All of these portals of entry are reinforced with heavy immune surveillance, heavy immune presence like we've talked about before. But we'll spend a couple slides talking about how these common bacterial pathogens can circumvent those defenses. Also, you don't have to know, by the way, these different pathogens. You will in the next couple blocks when we start talking about them in detail. But for now, these are just examples. Maybe some of them you recognize, and perhaps some of them you don't recognize, but just common examples of respiratory pathogens and gastrointestinal pathogens, urogenital pathogens. I just have 3 examples of classic sexually transmitted infections at the bottom there, but that's not to say that like E. coli, for example, wouldn't be one of those and others. So this is not by any means a comprehensive list, just a couple of examples. So these are the ways in, and also these are the common ways out when you are contagious. Once these different pathogens have had time to establish an infection and you start shedding them. If you're coming in through the respiratory tract, you're usually leaving through the respiratory tract as well when you're infectious. Similarly, if you have like a skin infection, let's say it's like an open skin infection where you have pus and other symptoms of that being an infected area, that would be a contagious source. You're shedding through that site as well. Modes of transmission, how this stuff is shed, can be thought of in 3 different categories that we'll talk about a little bit. First, when you talk about contact transmission, this is usually very close proximity or direct touching, those mechanisms. Vehicles are usually inanimate objects or transmission that would affect large numbers of people potentially. And then vectors, we're usually talking about biological entities, usually insects, oftentimes insects, that are capable of transmitting those pathogens from person to person. We look at contact transmission, a couple different subcategories here, direct and indirect. With direct transmission, this is usually talking about physical contact from one person who's infected to another person. Common ways of transmitting through direct contact would be things like shaking hands, kissing, etc., etc. Sexual contact is a great way not just to spread STIs, but since there's all of this going on too, you can transmit a lot of things through sexual contact. Pretty intuitive. What about auto-inoculation though? What is that? What does that mean? Yeah. Can you think of any examples where, I mean, if you already have it, then you already have it, right? Okay. No, I like that. I'm not going to repeat it, but good job. Yeah, you guys in the back. Say again? Yeah, so you said athlete's foot. That's a superficial dermatophytic fungal infection on your foot. If you have athlete's foot or jock itch or any other kind of like superficial fungal infection, you can transmit it to other parts of your body. So you need to be careful about touching it. That's a good example. Anyone think of anything else? Yes, sir. Yeah, exactly. I'd asked you guys earlier how many of you have had the smallpox vaccine, right? What do they tell you when you get vaccinated against smallpox? Yeah, patient education, right? Very important that you don't go in there with a vaccine and think that it's okay to touch it as it starts forming vesicles. Because that's a live virus vaccine. They vaccinate you with vaccinia virus. That's, so if you touch that and then you go touch your eyes, that's a primary concern. You can get a nasty, like vaccinia virus infection in your eyes in particular. Great, that's auto-inoculation, exactly. So, any other examples? Yeah. You can have unilateral pink eye and get bilateral pink eye real quick. So yeah, those are all excellent examples. Indirect contact transmission, if you've never heard the term fomite before, that's a good term to be familiar with. Anybody gone through field sanitation courses before? Not a single person. Okay, thank you. Do you remember what the 5 F's of field sanitation are that they teach you? Flies, feces, fomites, fingers, food. Fomites is one of those, and we'll talk about the other ones as well. But that's a basic tenet of field hygiene that maybe you've not heard it before, but basically it's that most of the things you're touching on a day-to-day basis, your cell phone, money, doorknobs. If you have kids and you're sending them off to daycare, those toys that they're playing with and sharing and slobbering all over, those are all fomites because they're inanimate objects that have some transferal of bacteria, usually sometimes viruses, and that when you hand that off to the next person, if it's soon enough where those organisms haven't died, you can transmit diseases that way. Now we don't use money much anymore because money is super dirty. But even bar soap, which you wouldn't think of as being a fomite, temporarily can be a fomite. When I was in Kenya, we had surveillance sites all over the country, and we'd travel out to these hospitals to do surveillance checkups. And some of those hospitals, like their bathrooms were set up where they're like outside latrines basically, and then you walk outside and there's this big long trough for patients and visiting family members to wash their hands. And so people don't steal this soap, the bar soap, they keep it in these cloth rags, these bags, netted bags, and then they attach it to the faucets at various points. And these, because they don't want people to walk away with them, but these bags that are keeping the bar soap are dirty. I mean, they're like discolored and nasty, and you do not want to wash your hands to those things. Even if they didn't, bar soap is far inferior to using like a liquid soap dispenser. You know, that's the way to go. Bar soap, if it looks dirty, it probably is dirty, especially if you are using it right after another person. Especially in a hospital condition like that, where you have lots of sick people concentrated in one area. Notice on this list of some examples, we've talked about ringworm. That can be transmitted by fomites too, like towels in the gym, for example. But I have common cold viruses here too. That's also up in direct contact, and it's also down here in droplet transmission. That just means that they're not mutually exclusive. You can get common cold viruses like adeno or RSV transmitted by multiple different ways. You don't necessarily just have to spit on someone in order to transmit it. But speaking on spitting on people, say it, don't spray it, right? If you've ever been super close to someone and you feel that little splash of moisture on your face when they're talking to you, if they're sick with something, you know, there's a good chance that some pathogen has just been directly delivered onto you. These are usually conversational distances less than a meter away from someone, where they can transmit some of those pathogens directly to you. Going on to vehicles, we'll skip down to airborne on that note. When we're talking about airborne respiratory pathogens, these are defined as distances greater than a meter. Some of these examples, most of these you've probably heard of, but these can linger in the air. See that figure there? These are kind of the ones that are small enough where they just hang out in the air. A lot of viral infections, for example, they can linger sometimes for hours, and some of them are shedding great quantities. An example of that would be like the measles virus and chickenpox. It's caused by varicella zoster viruses. These are both viral infections. And if you have somebody who has an acute infection and they're shedding a crap ton of viruses in a room, they leave that room, you walk into that room 2 hours later, you can get that infection because of how many particles are still lingering in the air. Heavily dependent on the ventilation in that area, whether or not the HVAC is working, but these are concerning because they do linger and they do travel distances and they can travel through ventilation systems as well. Waterborne and foodborne transmission, pretty intuitive stuff, just the stuff you're ingesting that is contaminated with usually enteric pathogens like Shigella, Salmonella, E. coli, Campylobacter, things like that. Some viral illnesses as well, like norovirus, some parasitic illnesses. With foodborne, unfortunately, some of these pathogens make their way into the food chain, like tapeworms can do that. And some of them are transmitted more through food handlers who aren't washing their hands, who are contaminated themselves. Like if they have hepatitis A virus and they're not washing their hands, they're doing food prep, that's been associated with big outbreaks of infections. With blood and other body fluids, these are pretty straightforward, but some of the agents you need to be cognizant of are the classic bloodborne pathogens like HIV, hepatitis B virus, as well as some of the more exotic ones. Creutzfeldt-Jakob, you guys remember what type of risk or threat that is? It's a prion disease, yeah, so that would be an example of something that can be transmitted through medical devices that have been used for surgical purposes in a patient who has a prion disease previously. Yes, ma'am? Prions, it could be, yeah, if you're talking about mad cow disease and eating undercooked beef. Yeah. And then finally, when we were talking about these insects, these insect vectors, we can stratify that into two different categories. The first is what we were talking about earlier when we were talking about field sanitation, that other F, flies. But it doesn't have to necessarily be flies. The reason why flies are commonly thought of is because they oftentimes, they'll eat anything, they'll land on anything, right? So they'll land on feces in a latrine. And if that fecal matter has an enteric bacteria, for example, like Shigella or Salmonella, those bacteria can adhere, like mechanical adherence to the outside of that fly's body and then be carried on to your food when they land on your food. So you can contract some of those enteric pathogens that way, by flies and other things like that. The other category of biological vectors, these include mostly biting arthropods. This is something Major Drury could probably talk about extensively as well, right? But with all these different types of biting arthropods, if they've evolved to carry these different pathogens, which are pretty well known, things like Yersinia pestis, and malaria, and yellow fever, dengue, a lot of these other vector-borne diseases, all of these pathogens need to have evolved in order to be carried in the unique environment of that insect or arthropod. What would you guess is the body temperature of a mosquito? Not 98.8 or 98.6. It's just lower than human body temperature. So that's just one thing, but in order, like human adapted pathogens must be able to survive at our body temperature of 37 degrees. But if it's a vector-borne disease or pathogen, those pathogens must also be able to survive at the lower temperature of an insect and with all the other environmental cues and signals that are different in an insect vector. So it is different than the mechanical vector carriage that we talked about here. Usually they can be carried for a considerable period of time in these arthropods as well. Yeah. Okay, so we've gotten into the body by one of these methods of transmission. We're talking about exogenous bacteria or even endogenous bacteria, but mostly focusing on exogenous pathogens that have gotten into the body. Now we need to establish an infection. This is where time becomes an important variable, because no matter how you get into the body, you're going to be confronted with the host immune system kind of right off the bat, right? This is where you have heavily reinforced immune surveillance. So not only immune surveillance, but you also have physical barriers, chemical barriers. You have things like flushing and peristalsis, all of these things that are stacked against you. So job one is to attach to something and hang on for dear life. If you are a bacteria that's capable of causing a urinary tract infection, if you're not able to attach to those epithelial cells lining the urinary tract, you're going to get flushed out immediately by the flow of urine, right, or pretty quickly before that patient voids next. And similarly for a number of other types of infections. So in order to attach to these solid layers in the body, bacteria will express things like pili and fimbriae that we've talked about before. Those short appendages that extend from the bacterial cell. And on the surface of those pili and fimbriae, they have these proteins called adhesins that act to facilitate the attachment of bacteria to whatever substrate they're trying to attach to. There are many, many different types of adhesins. This is just one example that's expressed by a couple different bacteria, but Staph aureus, for example, will express this adhesin that has a horrible acronym. It's MISCRAMS, microbial surface components recognizing adhesive matrix molecules. But I did not make that up. What those particular adhesins do is they allow the bacteria to bind to the ECM of tissue. So the ECM is the extracellular matrix, things like laminin, collagen, cadherins, things like that. And this picture is kind of showing Staph aureus cells, which are normally cocci, nice and spherical, but when they're attached firmly to some substrate, in this case it's artificial, it's a magnetic bead, but same. They flatten out because of all the adhesins that are making those tight connections with that magnetic bead. And that allows them to very firmly stay in place and resist things like peristalsis and flushing actions, blood flow if they're trying to attach to capillaries or heart tissue or things like that. Another way of establishing infection that's not necessarily mutually exclusive is the ability to invade tissues or sometimes be intracellular altogether. In most parts of the body, pathogens are going to need to be able to handle viscous fluids. A lot of the body is made up of viscous fluids. So for that reason, if the bacterial cell has agency to move away from threats or move towards nutrient sources or whatever, it behooves them to have some sort of motility. And that's what things like flagella enable them to do. It enables them to move away from threats or move towards nutrient sources in viscous environments. Not all bacteria are flagellated. Some parasites are flagellated, so flagella are unique to bacteria. But the gist is that motility can be a big advantage for some of these pathogens to maneuver around the body. Another facet of invasion is tissue digestion, particularly with bacteria. Some bacteria will produce a number of invasive enzymes that we'll talk about in a couple slides. But those invasive enzymes digest tissue. They digest fats, they digest proteins, proteases, lipases, they digest carbohydrates, hyaluronidases, all of these things to help cut through tissue and establish infections. And lastly, we have the ability to actually penetrate into cells and become intracellular pathogens. If you're a bacterial cell, why might you want to go into a cell and become intracellular? Yeah, like what's not, what are you going to be protected from if you're inside like an epithelial cell?
Antibiotics.
Antibodies, yeah. Intracellular pathogens have a good advantage in that the humoral response is usually not quite as effective for intracellular bacteria. So if you can escape like this bacteria, you can see it's being phagocytized. Some of them have evolved to want to be phagocytized. They actually elicit phagocytosis. So they're turning a defense mechanism that we use to clear them against the host, inducing phagocytosis because they've evolved some sort of defense strategy to survive what would normally kill them. If they're being induced, or if they're being phagocytized by a macrophage, how does that macrophage destroy bacteria? The lysosome, yeah, the phagolysosome, it has all these really potent antibacterial chemicals and has a low pH, super acidic and reactive oxygen species. So they have to have some sort of way of countering that in order to do that. But if they're able to do that, the benefit is escape from the immune system, which is a huge benefit. And parasitosis, if you've never heard that term before, that's simply darting from cell to cell. So you don't have to, if you're able to invade a cell in the first place, let's say you're an enteric pathogen like Shigella. Shigella can infect and invade intestinal epithelial cells, and it can like directly infect adjacent cells next to it. So it doesn't have any, the sun never shines on pathogens like Shigella. There's no immune, or very little immune exposure. And in terms of when you start talking about massive tissue damage caused by bacterial pathogens in particular, that allows you to have secondary infections and makes you more vulnerable in general when you start compromising tissues and the way tissues are supposed to function. Lastly, we have colonization. A lot of our body is colonized anyway with commensals. That's not a big issue. Our gut is colonized, that's fine. We have a little bit of colonization going on our teeth, right? What's that called? Plaques, dental plaques. That's a biofilm, which generally speaking is not considered to be a good thing. And with dental plaques, that's not a good thing either, but at least that's probably not going to kill you, right? In general, colonization is fine if we're talking about commensals living in their appropriate specific niche, but what you don't want is colonization of immune privileged sites. You don't want colonization of medical devices or implants. So those are examples of things you would definitely want to avoid. And biofilms are simply a really robust community of well-established, well-colonized, usually polymicrobial communities. So not just the polysaccharides, but that's a combination of excreted polysaccharides from different types of bacteria, extracellular DNA and proteins. All of that will bind those communities to themselves and to whatever they're adhering to, whether it be living or non-living. So lots of examples of that. Cystic fibrosis associated with biofilms, periodontitis, plaque biofilms that can grow on the heart tissue itself, urinary tract infections can involve biofilms, lots of examples. And then medical devices, what are some medical devices that are associated with biofilm formation? Catheters, CPAPs potentially. With catheters, you can have IV catheters or urinary catheters. Heart devices, heart implants, shunts, all of those things can have biofilms growing opportunistically. And then in terms of biofilms, protecting the bacteria from antibiotics in the host immune system, how might a biofilm protect bacteria from antibiotics? Yeah, that's true too. So,
Covers its receptors.
yeah, I think both of you guys are hitting on that. One is penetration protection, so the penetrating power of an antibiotic is compromised when you have a massive biofilm. And the other is immune surveillance. You're talking about the ability of the immune effector functions to reach down, especially to the bottom layers of that biofilm. They not only have to penetrate other cells, but they have to penetrate that mix of extracellular material that's protecting it. This red outline box at the bottom, I think it needs to be read verbatim because it's stated really well what the problem is when you don't treat an infection in a timely way and you allow it to get to the point where you have colonization. The longer a bacterium remains in the body, the greater its numbers, its ability to spread, and its potential to cause tissue damage and disease. If it gets to that point, an even larger host immune response and inflammatory responses are needed to resolve the infection, and that, as we know now, can contribute to greater immunopathology, which you also do not want. That can be fatal in and of itself. So this is a game of time and making sure that if you're going to treat, you treat in a way that you do not progress to colonization because it's so much more difficult to treat at that point. Okay, let's take a 10-minute break and we'll pick back up and talk about virulence at 10:53.
So back to
That's it. That one, you do it.
Yeah,
Dayton, Ohio.
I can send
Exactly. Make the circle.
it was like too slow.
Yeah, it's.
Reusing the. Yeah, I don't know.
No, exactly. Yeah, exactly.
Hundreds.
I don't know what to do. So I like that.
Finally, closing that loop. 6 years later.
Yeah, it's like literally just like just the and then I was like,
Just with your team, back
Xbox, electorals, also do. New season comes out. It's Smile. You get so much better. Yeah. But it's still and they do better. No, you get a new one. It's the last one. No, this is the last one. Well, I just started watching. I'm only watching the last season. I'm started. I'm like, dude, this is Oh yeah. Well, and it's not even all of them. It's just the part where they don't know. They don't really know. Oh, dude. Yeah. That's how I nope, that's not.
Yeah. I was skeptical of that. That was so big, and I'm like, no, you know, the kids are gonna eat it. And they didn't. I ate all of it. It was all good.
You never had fast food?
We've been slicing.
You like Reese's, right?
A piece of that, and then sit down and watch it.
Yeah. I almost had
They were like $3, and then it's like 2 for $4. I was like,
What the heck? What was that? Remember when we just talked about that? I ain't not sure how good for your cholesterol.
King size. Huh? King size.
On brand, the name brand peanut Cheerio. That we'd always get the off brand, and it was never supposed to. Why was it good for your cholesterol? I guess they're bringing a heat blanket in. But we had talked about the house getting cold. Yeah. My study gets pretty chilly. And so Truly comes in and I have a blanket like up over my face when I'm scrolling like right next to me. You want a jacket? Like, no, it's nice. No.
Usually like
How fast Sabrina is.
it was like in review for a hot second.
Oh my god.
Dude, I love.
Is that Oakmont?
He typically
Dude, barely tapped it. Did you see the rough that they're going to be playing in? Green speeds are at 16. Yeah. Dude. Someone posted, they're like, what's going to be the winning score? And like, whatever Scotty shoots.
No, it's supposed to be
You have to play like perfect all 4 days. I would fucking non-play. Huh? Said I would non-play on every hole. And you have to play to be below the hole every. Like you have to know that you have to put up. Because if you're putting downhill, you're gone. So they showed one. Guy had it like a, so it wasn't uphill or downhill. It was like a archer. He putted it and lagged it, and it started just trickling down, and it ended up in the sand trap. I was like, dude, that's not even. Xander, they were talking to him, and they're like, this is why people watch the U.S. Open. They don't watch people to hit 370-yard drives. They watch it because they know it's going to be the hardest round, and they want to see people shoot 8-10 over. Scores are going to be in the high 70s, low 80s. She just 30 slots. There's no way.
This is the what's the word? Still viable.
Alright guys. Back up again. Can someone give me the more or less your definition of what pathogenicity is? It's a bit of a review. Got it. Say it. How icky is it? how, would that be like a degree of ickiness, would you say? That would be, yeah, that would be, okay, that would be more like where we're going with virulence, like the degree of ickiness. But going back to pathogenicity, it's a quantitative or a qualitative term? It's qualitative.
Qualitative.
Yes.
The ability to.
Yeah, exactly.
The ability to.
Exactly, yeah. Yeah, he said it's the ability to, the qualitative, can you cause disease or not? And that sometimes is context dependent. That's the other nuanced point I was trying to make in those 2 or 3 slides is you might be perfectly safe hiding out in the nares like Staph aureus or Streptococcus pneumoniae, but if you get transplanted to an inappropriate niche, an ectopic site, then suddenly you're no longer commensal, you are a pathogen. But the term pathogenicity still means qualitatively, are you able to cause damage in that context or not? When we talk about the virulence of a pathogen, it's related, but it's not quite the same thing. Now we're talking about the degree of pathogenicity. There can be avirulent organisms that simply lack the ability to cause disease. They don't have any toxins. They're not well shielded from the immune system, which can also be considered a virulence factor. So avirulent organisms like certain strains of E. coli just have no fangs at all. Can do nothing. Wild type is a term we talked about when we were talking about microbial genetics. You guys remember what that meant? Like wild type? Yeah, it's a normal state, au naturel. It is what it is. So your wild type genome, a wild type gene, or you could apply that to something like a pathogen or a strain of something, wild type strain. Relative to that wild type strain, if you are more dangerous, would you be attenuated or more virulent? Yeah, you would be hypervirulent. What about if you are less dangerous? Attenuated. Attenuated means weakened relative to wild type. Good. This is particularly important when we are talking about strains, oftentimes of the same organism. Different strains can have extremely different virulence profiles, different capabilities of causing disease. Again, that example with E. coli, K12 strain, fine to work with benchtop, avirulent, non-pathogenic. K1 strain, super pathogenic, highly virulent. 0157 strain of E. coli, super virulent, packs a lot of virulence factors. So, different strains of the same organism. In terms of how we measure this, I'm actually going to start off, let's skip to this slide that talks about LD50 and ID50. One measure of virulence is the relative amount of that pathogen it takes to cause disease or damage. We have 2 different ways of expressing that. One is the quantity or the dose required to kill a certain percentage of exposed vulnerable populations. That would be the lethal dose, LD. And because we're usually plotting this dose as we're plotting this ability to kill people as a function of the amount of organism that you're putting, the dose or the inoculant, we plot it on a curve like this sigmoidal curve. And we usually use 50% of the population because that is the most sensitive point on that curve. It's the inflection point on that curve where change is most sensitively measured. Not down here where you have more of a plateau, or up here where 100% of the population would be killed, 0%. It's at the 50% mark. So the y-axis here, usually we're just drawing a line right at the number of people, right at the point where 50% of the people who were exposed would be killed. That would be relevant and useful to use for organisms that can kill you, that can be virulent enough to cause death, but not all pathogens are concerning because we're worried about them killing us. Some of them just infect us, right? Somebody had said athlete's foot earlier. Are you going to die from athlete's foot? Trichophyton that's growing in your foot? No. But do you want it to be infecting you? No. So for that, there is a dose related to even something like athletes, but for that it'd be more appropriate to use an ID50, the dose that would be infectious for 50% of the population. All right. Looking again at this comparison, we have two different strains, so this implies that it's the same organism, just one is more virulent than the other. If we look at the dose between strain A, this red line, strain A is labeled up here, versus the dose required to kill 50% of people for strain B, we can see there's a difference. Strain A will kill half the people exposed at 30 organisms. In strain B, it requires 50 organisms to cause the same amount of death. Which one do you think is more dangerous? A, intuitively, right? But why? Somebody
It
articulate.
takes less concentration, less of that microorganism to kill the population, so it takes less, so it's more harmful.
Yeah, exactly. So it's a little crude, but it works, and we use this term all the time. And we use these terms to express the amount of concern or risk associated with different pathogens. One other point to make, you could do like an LD30. I've seen stuff like that. You could do an LD80, you could do an LD100, but LD50 is usually what you see. Okay, so again, that's more sensitive part of the curve. Now, let's go back to this slide, this is the earlier slide. That blue line in the middle would be our wild type. Similar to what we just talked about, let's say we actually have a dose associated with the LD50 for that wild type strain, and let's say that dose is 50 organisms. If you shifted that dose down to 20 organisms, would you call that attenuated or virulent relative to wild type? Virulent, yeah, so it's a little counterintuitive, but you're decreasing the required dose, and you have a more virulent strain. It's less that organism. Similarly, if you did something, let's say you're talking about Bacillus anthracis, which has a couple different plasmids that encodes things like capsule and toxins. Let's say in the lab you were to knock out one of those plasmids, like delete the plasmid that has capsule production. It's as soon as you get Bacillus anthracis into that host, the host immune system, those macrophages are going to come and phagocytize the non-encapsulated strains. It's going to be much, much less virulent. So a strain like that could have a huge increase in the LD50. Instead of 50 organisms, now it might take like 1,000 to kill 50% of the population. Or maybe there's no LD50, maybe it simply can't kill anyone. Okay, makes sense? Okay. This is definitely an FYI slide, but these are examples just for you to kind of sit and ponder at your leisure. Different viruses, different bacteria that have different infectious doses, not ID50, just the infectious dose in general. There's a whole bunch of qualifiers and specifics probably that went into generating these data, but in general, you can kind of get the gist of it, that it varies, and this is one of the reasons why we study pathogens separately and independently, because each one has different characteristics. If you skip all the way down to the bottom here, Vibrio cholera, causative agent of the disease cholera. This estimated dose at 100 million bacteria to cause disease, that might be true. It does take a huge inoculum to infect somebody with cholera. But that's wild type cholera, and that's from contaminated water. When you have somebody who's infected with cholera, they themselves, once they get sick with cholera, are infectious, and you can get person-to-person transmission. And then this environmental bacteria, Vibrio cholera, that lives out in environmental waters, can become human adapted, and it gets better at infecting people. And when it does that, it becomes more virulent. Each time it gets passage from person to person through the fecal-oral route, it gets more virulent, and this infectious dose starts dropping. So it becomes, instead of 100 million, it might be 100 times more infectious, or 1,000 times more infectious. So those are some nuances of this, but in general, these are some of the infectious doses of different organisms. All right, now we are going to spend a little bit of time talking about different virulence factors. We've talked enough about bowel inflammation, we still have a little bit more talking to do about, talking about immune defenses, so we'll talk about this a little bit more. Intracellular growth, Endotoxin and exotoxin, as a way of contrasting those two, we're going to hit hard because we haven't talked about exotoxins a lot yet, but we have talked about endotoxins. Talk about invasive enzymes. Not a whole lot of time spent on superantigens anymore. A little bit about metabolic products of bacteria that can cause tissue damage, and not a whole lot about host nutrient acquisition, just lightly touching on some of these. First, going back to the immune system, talked about this a little bit, but if you want to evade the immune system, one of the primary strategies that pathogens will take is masking themselves or shielding themselves from detection, things that will recognize the antigen on their surfaces. Some pathogens will do this by mimicry, where they they've evolved a mechanism where they literally will display antigens that look like something entirely else, so they won't be recognized. Some parasites do that. And other pathogens like influenza virus, very famously, is a good example of a virus that mutates. It's an RNA virus, but it mutates enough, and there's sort of like a preferential mutation for those glycoproteins on the surface, the ones that would most likely be seen by antibodies, to constantly change. So the immune system is constantly having to adapt to different antigens that are displayed on the surface. Another example of antigen with influenza specifically is antigenic shift, which I mentioned earlier. That was viral reassortment events, where you have two different strains of influenza combining and infecting a single cell and causing a shuffling of these different genomic fragments together, where the end result is something entirely new that has not been seen, that is not recognized by vaccines, and is certainly not recognized by your immune system. You don't have any memory T cells, you don't have any memory B cells. That's antigenic shift. Intracellular survival, we talked about why it might behoove some pathogens to find a way to survive this very dangerous process that you would encounter when you get engulfed and then enter into a phagolysosome, which should be designed to kill you. But here are a couple different examples of mechanisms where bacteria in particular have survived or evolved ways of surviving this. Some of them will produce enzymes that stop the phagolysosome from fusing together. And when that happens, they can eventually produce enzymes that will allow them to penetrate out of that compartment, that endosomal compartment. Either way, you're not going to stay in that lysosome forever. You're going to need to get out one way or another. So phagosome escape is going to be necessary as well. But once you escape from that phagolysosome, some of these bacteria can survive and replicate perfectly fine within that host cell and the cytosol. So as long as they've evolved one of those mechanisms to get out of that phagolysosome. No additional strategies for evading the immune system. Some of these strategies involve things that thwart antibody function. We talked about how antibodies help get rid of pathogens. They bind with their Fab region, right, the arms of that Y-shaped molecule to the pathogen, and then the Fc portion is the part that gets cleared, usually by some immune cell that contributes to that. This first example here is actually the bottom bullet. This is related to bacteria like Staphylococcus aureus that produce a protein called protein A. And when staph produce this protein, they start coating the exterior of their cell wall with protein A. What this does, what protein A does, is it binds to the Fc region of those antibodies with super high affinity, like incredibly high affinity. At that point, the antibody is turned around in the wrong way, right? The Fc region is now bound. So all the effector functions that are conferred by the Fc region are negated in that process. You just kind of pointlessly have the Fab reaching out into space, not able to bind anything because the Fc region is bound to that cell. Okay. Antigenic masking we've talked about before. That's something that can be facilitated, for example, by capsule production. Those bacteria masking the antigens. So the antibodies cannot recognize them. And another mechanism of thwarting antibody function is producing these enzymes that can, these proteases, that can actually cleave the antibody molecules right there at the Fc Fab junction where I drew a line across there for you. Those anti-IgG proteases, there's also anti-IgA proteases as well. When they are cleaved like that, the antibody is completely useless. Cannot do anything. It's just a Fc region floating around or Fab region floating around. I did add in there too a little bit of extra text for this protein A-B function. It's not just complement activation, but any other Fc-mediated clearance mechanism would also be negated by that. So, inhibition of phagocytosis. We have a couple other examples of virulence factors that certain bacteria express in order to not be phagocytized at all in the first place. So this would be, we talked about professional phagocytes before. What were some of those immune cells that are really good at phagocytizing? Macrophages, dendritic cells, those are primary phagocytes in the immune system. Okay, so we're talking mostly about those cells. Capsule, you'll hear this again and again and again because it's really important. Capsule-producing bacteria have a much easier time not being phagocytized. Now we're talking about another protein, M protein, the second bullet, not to be confused with protein A on previous slide. M protein is a completely different protein. It's produced by Streptococcus species. M protein is also expressed so that it covers the surface of that bacterial cell, but has a different function. Protein A will bind the Fc region of antibodies. M protein does not interact or bind to antibodies. That's one big difference. What it does bind to are serum factors, like serum factor H, which ultimately stymies the ability to activate complement. So ultimately, these bacterial cells, which would otherwise be opsonized by complement factors like C through B, will not be opsonized, and that makes it harder for phagocytes to engulf them. It just messes up the opsonization from complement. Coagulase, that's what we're seeing here in this figure, the action of coagulase. What coagulase does is it stimulates the formation of clots intentionally around those bacteria. Why would a bacteria want a clot around it? Exactly like it says up there, to prevent it from being phagocytized. It's a play for time. You can't be like that forever, but if you just need to buy a little bit of time and not be phagocytized, you can secrete coagulase, which some Staphylococcus species do, coag-positive staph. They will do in order to escape being phagocytized. They just need to have a mechanism to break down that clot at a later point in time. But leukocyte production, it's exactly like it sounds. Something that kills leukocytes, causes the destruction of white blood cells and leukocytes. So it does so by poking. It's a bacterial factor that pokes holes in leukocytes and destroys them. Inhibition of that fatal lysosome fusion is another mechanism. And then some bacteria have ways of inhibiting chemotaxis, so you don't have those phagocytes being recruited into that area in the first place. These invasive enzymes, I know this is kind of like an obnoxious list, but I did warn you that we would have some, material where you just kind of have to memorize it and know what it does. Fortunately, for most of these invasive enzymes, the function of these is written into the name, so as long as you understand what the name means, you can remember what it's actually doing. These are the enzymes that contribute to a lot of the tissue damage that we were talking about directly. The bacteria that produce these invasive enzymes can cause some really serious infections, things like necrotizing fasciitis, a lot of extensive damage directly from the bacteria themselves. Collagenase breaks down collagen fibers. The cypinase is a phospholipase. It breaks down parts of that, the host cell membrane, destroys them that way. Hyaluronidase breaks down hyaluronic acid. You don't need to know these in any more detail than what I'm going through right now, just stuff that's on the slide. DNase is a little bit trickier. With DNase, these are enzymes that do degrade DNA, but they're the DNA nets that are thrown by those neutrophils. So some bacteria Netosis is so common that bacteria have evolved DNase expression as a way of getting out and surviving netosis. So DNases will destroy that extracellular DNA that gets thrown at them. Other bacteria will find ways of breaking through blood clots when they're ready to disseminate and spread throughout the body. So things like fibrinolysins and streptokinases. Streptokinases are just like a type of fibrinolysin produced by Streptococcus species. All of those will contribute to degrading fibrin clots around them so that they can spread and continue to invade tissue. Hemolysins, what does the name suggest they do? Lyse red blood cells, exactly. Lipases break down lipid membranes, and then proteases, there's whole classes of proteases. Some of them will do what we talked about the last slide, cleaving antibodies, and some of them will do other functions that destroy host proteins. All right, getting into toxins. Again, endotoxins, you guys have had a pretty good primer, but there's some information here that you might not be familiar with. The point here is you need to be able to differentiate between endotoxins and exotoxins. Starting off with what the names imply, the prefixes endo versus exo, you should be able to parse out what exotoxins are relative to endotoxins. What are endotoxins?
They attack the outside of
Do they attack anything?
It's just like toxic waste.
It's not waste. It's
Protein.
go ahead. Yeah, he said it's an integral part of the outer membrane of gram-negatives. That structure that we've seen in Block 1, the structure you need to kind of have in your head for that exam on Monday, what that structure of the outer membrane of gram-negatives is. You have in LPS, which is that layer of this outer membrane, extending from those lipopolysaccharides on into that core, the core oligosaccharides, and then deep into that actual lipid A layer of the outer membrane. This is the endotoxin part. If it's lipid A, that's what's it composed of? Is it protein based? It's lipid. Yeah, and then the entire LPS also has saccharides involved, but the lipid A portion, that's primarily lipid based, it's not protein. With exotoxins, we are talking about proteins for the most part. So structurally, proteins versus LPS, in terms of where endotoxins are themselves, this is integral, as he said, integral to the outer membrane of gram-negatives. Endotoxins, integral. Exotoxins, secreted outside of the bacterial cell wall for some effector function. Exotoxin. There will definitely be some questions about this. So, yes, sir.
Would you then say that exotoxins are proteins and endotoxins are more lipids?
Mhm.
Just generalized? Is it like an always, or is that like? Would that be an easy way to break it down? Say exotoxins are more proteins, and then endotoxins are.
Yeah, in terms of composition on this slide, most exotoxins are primarily, like they might be like cosylated potentially, but they're not lipid based. Endotoxins, this usually isn't going to be a big problem. It doesn't usually stimulate an immune response because of the fact that it's embedded in that relatively inaccessible part of the outer membrane. This is what's seen more by the immune system, that O antigen. The lipid A part is not visible as much. So when it becomes a problem, it's usually when those bacteria are dying and this is suddenly exposed and creating all sorts of problems for the immune system. TLR4 activation, things like that. Okay. So if you understand that part, then a lot of these other compare and contrast bullets make more sense. So let's keep going down. Composition, peptides versus LPS, lipid A portion. In terms of stability, those endotoxins are lipid-based, so they're going to be as stable as that bacterial cell is. They tend to be more stable than exotoxins. There's a bit of a disclaimer on that. Some exotoxins are thermal stable. Most of them are thermolabile. but generally, most of them, again, will be destroyed by heat. In terms of immunogenicity, this weakly immunogenic bullet for endotoxins, that is relative to whether or not the bacteria are intact. If they're intact, they will not elicit a strong immune response, or as strong as if the bacteria were lysed or destroyed, and they become immunogenic. With exotoxins, this is one of the reasons why the humoral response is so good, because antibodies recognize these protein-based exotoxins that are secreted. So they're highly antigenic, highly immunogenic. We have vaccines based on toxins that we've developed against toxins, which brings us down to this next point there in the whatever 6th row. In terms of developing vaccines, you mentioned DTaP and Tdap. What kind of pathogens do those vaccines protect us against?
Tetanus, diphtheria, pertussis.
Diphtheria,
Pertussis.
pertussis, exactly. So those are toxin-based vaccines for toxin-based damage from toxin-based pathogens. Okay, when they're synthesizing and developing those vaccines, they're growing up the toxins themselves and oftentimes deactivating them with formaldehyde, so they're no longer potent, but they can be used in that vaccine to stimulate the humoral response. So you raise antibodies against tetanus toxin, and you raise antibodies against pertussis, or diphtheria. Regardless, the point is, they're immunogenic. So they're useful for developing toxoid-based vaccines. In terms of potency, these tend to be highly toxic and very potent, so small amounts on the order of microgram quantities or less can be fatal. Endotoxins aren't nearly as potent, so the dosage is much higher to cause damage. Another important point with exotoxins is when bacteria like diphtheria or Clostridium tetani or some other toxin-based bacteria are producing them, those exotoxins require a specific host cell receptor. A diphtheria toxin is not going to be broadly toxic to a bunch of different cells. It's going to be toxic to cells that have that a receptor expressed on the surface. They have specific receptors for them. And that's a trait for almost all exotoxins. They have specific receptors. Endotoxins, again, you get the release of endotoxins out into the body. All that's stimulating is a non-specific response, pattern recognition from PRRs and toll-like receptors, basically. So non-specific. Or specific receptors just being toll-like receptors, but it would be damaging broadly, not specific. And then in terms of symptomology, exotoxins usually do not produce fever, at least not directly. The bacteria themselves may, but they will not usually produce fever. Endotoxins do because you have stimulation of and release of pro-inflammatory cytotoxins like IL-1 and IL-6, tumor necrosis factor alpha. And then lastly, where they are produced in these bacteria, with exotoxins, these are usually going to be on extrachromosomal genes, read plasmid-based encoded. Remember when we were talking about plasmids and what kind of genes they encode? A lot of them have virulence factors. Virulence factors like toxin production.
The bells and whistles.
The bells and whistles, the nice to haves, the frills, if you will. So yeah, plasmid-based. Not always, but oftentimes. With endotoxins, again, like you're talking about a lipid in the first place, so you're not directly going to express genes that directly synthesize lipids. You have to do that with proteins that construct lipids and make them into the structures and end up getting put into those membranes. So it's a different synthetic pathway altogether. And it's essential too. You're talking about the outer membrane of bacterial cells, so that's not a nice to have, that's a must have, right? It's integral. So that's definitely going to be chromosomal. We talked about endotoxins enough, but with exotoxins here, this is an example of a couple mechanisms that bacteria have of getting those exotoxins out into target cells. They do so by expressing these very complicated macro machines called type whatever secretion systems, type 1 secretion systems, type 2 type 3, type 4, type 5, type 6. There's at least 6 different types of secretion systems. So these can have a dozen or more different proteins working together in order to move whatever toxin that bacteria is producing from it to a target cell. Some of these secretion systems, like on this slide, it has type 3 and type 6. Will move that directly into the target cell. It will form a pore and actually inject it into that target cell. The other example is a type 2 secretion system, which is moving those exotoxins out into milieu, the extracellular milieu. So those exotoxins are on their own to find a cognate receptor and do that damage. But either way, this is the delivery mechanism that bacteria have for moving those exotoxins out. This is just kind of like a protein model of what these very famous anthrax toxins look like. Leak factor and edema factor that anthrax produces. Just protein-based exotoxins. This slide, I hesitate to even show you. You do not have to memorize it. The point of this chart is just to let you know that these different major exotoxins have specific effects on specific types of target tissues. That's all. Say again?
So it's an FRS.
The examples is an FYI, and with this, you would not be required to memorize this, but again, the point, you need to know the point of the slide. It's supposed to be reinforcing this bullet that you have a specific target action. We will definitely not go through all this in detail, but we will go through a lot of this when we talk about these specific organisms, like when we talk about Corynebacterium and diphtheria toxin. We might go into more detail about how that toxin works, like what it's targeting and how it's working. With Staph aureus, we spend a lot of time talking about Staph aureus because it's such a major pathogen. So we'll talk a little bit about some of the exotoxins it produces and what it does specifically to start degrading tissue and causing some really terrible skin infections. So you do not need to memorize this slide. Okay, just know that they are specific actions. This is sort of a summary slide showing you what is possible when you have the full complement of a highly virulent bacterial strain that can cause incredible damage to the body. These would be examples of things that bacteria that are producing invasive enzymes that are breaking down tissue. They are producing gas and metabolites that's causing tissue damage. This MRI here, you can barely see it, maybe you can see it on your screen. But this is gas production in the middle of somebody's cavity. This MRI is showing there's metabolic byproducts produced by Clostridium perfringens, which is a really rapid growing anaerobic bacteria that can cause necrotizing fasciitis. So like when these bacteria are rapidly dividing, they're dividing so quickly that you can actually hear gas moving through tissue, like there's an auditory sound associated with this, horrifying. In terms of what's going on here, you can see a lot of immunopathology too from the host. There's a huge swelling, lots of edema going on. You can see those sutures are like being ripped as that tissue expands. This is some really infected tissue. At the bottom here, you can see devitalized, devascularized tissue. This is a patient's back. So I think it started with a decubital wound that festered, got infected with Clostridium perfringens, and all that tissue is necrotic, it is dead, it has no oxygen coming to it, all the vasculature has been destroyed. So yeah, it's rotting, that tissue is literally rotting, here too, rotting tissue. Here you can see another type of necrotic infection. This is caused by Streptococcus pyogenes, group A strep, we'll talk about this as well. And here, again, like the tissue damage is so extensive. Before they debrided it, this is what the patient's leg looked like, and you can see there's massive amounts of swelling. But that still doesn't tell the whole story of all the damage that has happened underneath the surface of the skin. When they started to debride that tissue away, that infection went deep, and that necrosis went deep through the skin, through the fascia, through the fat, into the muscle. Sometimes it can go all the way down to the bone. Sometimes debriding is not enough and you have to amputate. So all this is to say you get some really virulent strains that are producing a lot of exotoxins. There can be a lot of tissue damage, a lot of invasive enzymes, and then immunopathology all working together to cause disastrous effects. That's a great question. I don't know. The docs who treated this patient, this wasn't in the United States, but they wrote a case report and they published it in JAMA or some other publication like that. I don't know what his outcome is. But yeah, these diseases generally have a fairly poor prognosis when it's that extensive. Yeah. And sometimes you can't really debride when you're talking about tissue. That's it. I don't know. But yeah, poor prognosis. All right. A couple slides about host defenses. This is just pure review. We've been through this before, but this is going back to the basics that we covered in block 1, just to remind you that you still have those natural barriers for infection and then primary protection, secondary line of defense from innate responses, and then third line of defense from adaptive. That's from the host, and you also have contributions from the normal microbiota as well, just to protect you. You have competition from bacteria that are inhabiting these different niches. That is helpful. They might secrete their own antimicrobial peptides to damage competing species, or they just simply crowd those other bacteria out of the space. But those are important functions as well. The image of the Japanese subway system, this is in Tokyo during rush hour, that was supposed to be an analogy to crowded conditions, and the fact that when you have a crowded niche like that, it discourages other bacteria from occupying that niche. It is simply like, would you want to get on this train? It is too crowded. So. Okay, I've got 11:33. Do you guys need a break? Okay. All right, we'll push on. The last 10 slides or so of this deck are epidemiology, important stuff, but we can go through it, I think, fairly quickly. Some of these we've already talked about a little bit. Remember when we were talking about portals of entry and how those portals of entry, like the mouth and the airways, etc., they can also be portals of exit. This is called shedding, and this is after you've had, after those pathogens have had an opportunity to start replicating to quantities where they can be shed from that host in infectious quantities when you become contagious, in other words. This is an indication of transmissibility. Not all organisms are shed in huge quantities. Those that are shed in huge quantities are generally considered to be more virulent, and they certainly are associated with outbreaks. Norovirus is a great example because norovirus infections result in diarrhea, basically, right? The type of diarrhea that's caused by norovirus is oftentimes described as explosive diarrhea. So you have an event and it aerosolizes, and these aerosolized particles of fecal material are airborne. They can travel huge distances and infect a lot of people. So you get norovirus, yeah, no shit. You get like huge norovirus outbreaks that are associated with crowded conditions, like on cruise ships, famously norovirus outbreaks on your super expensive Carnival cruise, in daycares where kids are whatever. Anyway, that underscores the point. If you have an explosive diarrheal situation, stay at home. Hopefully you live alone. Hopefully you just go somewhere and be alone for a while. That's what needs to happen, because that stuff is more infectious than you might think. It travels really far. And we give a lot of attention to respiratory viruses and respiratory infections, but don't underestimate the outbreak potential of diarrheal disease. Other major pathogens are major for other reasons, but they're not that good for transmitting disease. Mycobacterium tuberculosis is one of those examples where even in an active case, you're maybe not shedding a huge amount in sputum. And another example, Mycobacterium leprae, another species in that genus, that really is a low shedder. It's very hard to transmit. What does that bacterium cause? Leprosy. Yeah, exactly. Very difficult to transmit leprosy, or the bacteria that causes leprosy. Shedding volume depends on the time course of infection and sources of shedding. These are fairly intuitive. With the fecal one, going back to that, just to close the loop on that, fecal oral transmission is exactly what it sounds like. It's gross, but it happens. You have all of these enteric pathogens, whether they're viruses or bacteria, that are able to continue transmission cycles because we don't have sufficient hygiene to prevent contact with fecal matter. That's what happens with all of these fecal oral transmission mechanisms. One way or another, that fecal matter unfortunately ends up usually in your food or your water or on your hands through fomite handling, and then that goes into your mouth. That's fecal oral transmission. Or more direct methods. I was watching my cat groom himself the other day. Direct fecal oral transmission. I have to say that. Like he was just demonstrating how that's done. So, fortunately, never mind. Moving on. When we're looking at the time courses of disease and talking about shedding and transmission, it really does all come down to the pathogen specifically that you're talking about. Another reason why we take the time to talk about these pathogens independently and separately. The example I have up here, I have a couple of examples. Influenza as a paradigm for viral infections is good, but the timeline down at the bottom is showing COVID. But they are different. They're similar but different, close enough. But you can contrast that with a bacterial infection like Salmonella, and you can start seeing that there could be appreciable differences in when shedding might occur relative to symptom onset. So let's look at this in a little bit more detail. We'll just look at the COVID one so we get a sense of what some of these terms actually mean. When we're talking about an incubation period, we're talking about the period from the time you get exposed to COVID or influenza or Salmonella to the time of symptom onset. For viruses, that could be a relatively short amount of time, but for influenza, incubation period on average 5 days. But, it could be 4, it could be 6, whatever. Some pathogens can take years as an incubation period. Mycobacterium leprae is another example of that, where the incubation period is extremely long, like years. Nocardia is another one where it can take months, potentially. And then after that period, after that incubation period, if you've had a successful infection and the infectious dose is sufficient enough to overcome your initial innate response, you start getting replication, then you oftentimes get symptom onset. With viral infections like COVID, for example, even with a mild infection, the main point here is that you can be shedding virus before you are even symptomatic, which has implications for transmission, doesn't it? You don't even know you're sick. You didn't know you were exposed, you don't know you're sick, and you're already shedding virus a day or two before you become symptomatic. When you become symptomatic, then you should know for sure at that point you need to stay at home, wear masks. But up to that point, you have no idea. Okay, so that's important. With a bacterial disease such as Salmonella, oftentimes you become sick, sometimes because of immunopathology, but you become sick before you start shedding. Salmonella is an example of something that we shed in fecal material. Other points here with mild COVID, as you stop shedding, you could still be undergoing, even though you're no longer infectious, that inflammatory phase where you have immunopathology and symptoms related to COVID, they can extend even longer past that actual infection. And then hopefully you will recover at some point. With severe COVID, recover or die, depending on how that infection goes. A couple other terms related to epidemiology. One we use a lot is the attack rate. attack rates are useful when we're talking about a defined period and an actual outbreak. Also, by definition, this is the proportion of exposed and at-risk people expected to be infected. It can be a tricky term to use because we do have so many vaccines, and when we vaccinate routinely against a pathogen, we're no longer really considering that population to be at risk or vulnerable anymore. So take, for example, measles. If we are trying to hit 95% vaccination rates, that's not really an exposed population anymore. So it becomes difficult to calculate an attack rate. A better example would be something like Ebola, where we don't routinely vaccinate against Ebola virus. So it's a pretty at-risk vulnerable population. Then you could calculate an attack rate for an outbreak. How that's done is simply the number of new cases divided by the people at risk. You can just leave that as a fraction or make that a percentage multiplied by 100. But at the bottom here, let's kind of walk through these examples. We have mock-ups of an infected person transmitting to uninfected people and getting them infected. So we have scenario 1, 2, and 3. If you had to calculate attack rates for these 5 people who are exposed to an infected person, what would you say for that first one, where this guy is transmitting to 3 out of those 5 people? 60%. 60% attack rate. The next one, he's only infecting 2 people. 40%. And then the last one, everyone who comes in contact with him is at risk of getting infected, so the attack rate is 100%. Reproductive term is another term we use in epidemiology sometimes, but it too has a lot of limitations. With reproductive number, we're also talking about a completely susceptible population most of the time. I think the waters got muddied a little bit with COVID because they tried to model this based off of populations that had some protection from masks or social distancing or vaccines. But where it's most useful is a completely susceptible population. This is the average number of secondary infections expected to arise from a single infected individual in that population. And it gives you an indication of how infectious it is or how transmissible. Here, if we have an R naught, that's what the R subscript 0 means, R naught. An R naught of 0 means that that infected person will not be expected to transmit to anyone. An example of that is rabies virus, where unless you're fulminant rabies and biting someone, you're probably not going to transmit to anyone. So rabies usually has an R naught of 0. No human-to-human transmission, very rare. An R naught of 1 is pretty crappy for the pathogen itself. It means that it can only, on average, be transmitted to one other person. Anything between 1 and 0, if you have an average R naught of like 0.5, that outbreak's going to die out statistically. It will not be able to sustain itself. Those slowly just fizzle out. And technically, anything around 2, that outbreak is going to continue on. So every infected person, on average, is infecting 2 more people. That's an outbreak that can grow, theoretically. Before I move on to case fatality rate, I'm just going to skip to this next slide that talks about R naught examples, and I'll come back to CFR in a second. But when we talk about the R naught, the most famous example is measles. It's given as the most transmissible, just about the most transmissible infection we have. It has an R naught of 12 to 18. It's airborne. It lingers in the air after an infected person is left. And for every infected person, that infected person can take up to 18 people down with them if they're not vaccinated. So extremely infectious, very transmissible. And at your leisure, you can kind of mull over some of these other viral infections. I think these are all viral. Yeah, these are all viral. And kind of examine the R naughts. But most of these we do have vaccines against. We do vaccinate against them. So these R naughts are a little bit squishy with some of these. Ebola, terrifying disease, very high case fatality rates. I'll get to that in a second. But not that transmissible. It requires contact with blood and body fluids to transmit Ebola, not airborne. More difficult to transmit. So with case fatality rates, this is the proportion of infected people who die from the disease during a specific period. This is not like in general, you don't have a case fatality rate in general. You have it for a specific outbreak for the time period. And that gives you, when you're talking about outbreak to outbreak, it gives you an indication of how serious that outbreak is. CFRs for different types of Ebola, like Zaire or Sudan, which are considered to be more virulent strains of Ebola, are much higher than outbreaks caused by less virulent strains of Ebola, like Lucifer virus, for example. So, useful in those applications.
So, R naught, is that correct? Is it safe to say anything over 1 equates to exponential growth, or like, you know what I mean, like spreading? Like, so 1 is persons, it can only affect 1 person at a time.
Yeah, on
Is-
average.
So anything over 1, or is it kind of jump to like, if it's not up to 2, then you're not going to get that exponential?
That's a good question. I kind of, this is not a great figure to show exponential rounds of transmission cycles, like 3 or 4 transmission cycles deep, where you start getting an appreciation for that. 1, yeah, you certainly will not grow an epidemic with just R naught of 1. R naught of 2, yes, you could, with a couple transmission cycles, get a lot of people infected. But when you start getting into these higher R naughts, like, I don't know, mumps on up, and certainly measles, then you can have some really dangerous exponential outbreaks. Back before when smallpox was still ripping through the population in North America, some of those outbreaks were enormous. A bacterial example that is not on the list because this is all viruses is plague, Yersinia pestis. Some of those outbreaks have very high R naughts, very transmissible. There's absolutely nothing people could do to avoid getting the plague. You had a question? Okay. But does that answer your question further? Okay. All right, herd immunity. This is a concept we've touched upon before when we were talking about vaccination and immunization in Block 1. So most of this, I think you know, the intent is to develop those memory cells in the adaptive immune response, humoral and cell-mediated arms of the adaptive response. You do want to hit that target percentage of the population. If you get under that, you do not get the effect of population-wide protection. It's another major theme here. But importantly, it reduces the number of people who are susceptible in that population as a point of herd immunity. That leads to fewer new cases, lower transmission rates from person to person, and reduced microbial burden in the community. Really good example, I won't talk about it too much today, but that measles outbreak that happened earlier this year in West Texas. That particular community was well below the national target average of 95% immunization. They were like 12 percentage points lower than that. And because of that, you were able to have transmission events because people were susceptible in that community. This last point too, I think is super important. It's something that you would think we would kind of understand better when we have really severe outbreaks, but during COVID, before we had pharmaceutical countermeasures, before we were working on a vaccine right away, we knew that that was going to be our way out. But before we were able to develop that vaccine and medical countermeasures and antiviral treatments, the only thing we had were non-pharmaceutical protection methods. So public health was trying to get not just the American population, but the world population, it was a global pandemic, to adhere to these basic mitigating strategies to limit transmission cycles. And we couldn't do it because people couldn't stop thinking about themselves separately and independently. They didn't see the bigger picture of transmission. But before you have pharmaceutical measures available, you have to rely on non-pharmaceutical protection in order to limit transmission cycles. Yes.
And for the herd immunity percentage for all, like, is it like, is it 95% for each condition, or does it just depend on?
It depends on the disease. For measles, it is set at 95%. That's what public health experts and epidemiologists have calculated for that virus based on the R naught and based on the vaccine efficacy. It might be different for a different virus that has different transmissibility factors. Like it's certainly a different for smallpox, for example, like we no longer have a target or a goal of having herd immunity for smallpox in the general population of the United States. It's no longer a goal. So it does vary. Okay, for epidemiology, just to close out, I think this is our last slide on epidemiology. Couple considerations when we're talking about protecting the population writ large. One is that these infectious agents are often highly adaptable. On that last slide, we saw a bunch of different viruses. Viruses mutate at very high rates, especially RNA viruses, which most of those were RNA viruses. But even bacteria will mutate. In all of these cases, if you are working on either new drugs or vaccines, you have to stay abreast of those changes, and that requires surveillance methods. So surveillance is absolutely critical. You need to have information on what is circulating and how those pathogens are changing. This takes a lot of money, takes a lot of effort, but it is necessary. Available medical expertise is not always applied. This was big when we saw vaccine hesitancy here in the country about COVID vaccines, but also it is more understandable in other parts of the world too, where vaccine hesitancy is relatable, because governments have failed people before, and why would they trust a vaccine? So it's understandable a little bit why some people might hesitate to get vaccines. Plus, science is not infallible, is it? We have had crappy vaccines. Not all vaccines are equally effective, and some of them have caused problems. It's been public health disasters when that happens, but it does happen. Even if people are willing and seeking vaccination, not all of them have equal access to those vaccines. Too, remember back in COVID when some of those lower income countries were trying to get the vaccine and they were queuing up, and it became like a big political hot button issue, how to get access to COVID vaccines. So it took a while, we eventually did. HPV, that was also an access issue in some cases, but heavily politicized. You guys know what HPV is?
You mean papillomavirus?
And the vaccine that we tried to make available in 2006, Gardasil. Yeah, what happened with Gardasil? Trying to roll that out and offer it to the public. Yeah. Oh, no, not medical issues, but like what more psychological? What was the big? What ages were we offering Gardasil? Yeah, and even younger, like 12-year-olds, preteens. Parents were freaking out. They didn't want to even imagine that this virus, which is designed to protect against forms of HPV that could be transmitted sexually, they didn't want to think about their kids being vaccinated against an STI. So there's a lot of resistance there. Differences in changing social norms is another thing that we can consider when we're talking about managing population level public health threats. A couple of examples there. Before we had air conditioners, we hadn't characterized, we didn't know the existence of a bacteria called Legionella pneumophila. Legionella pneumophila is an environmental bacteria. It does well in water, and it really wasn't so much of an exposure risk prior to air conditioning systems being widely used. We started air conditioning buildings around the 1950s. That became popular, and it only increased in popularity over the last 80 years or so. But what this pathogen is able to do is, particularly when you have faulty HVAC systems or faulty air conditioners, it gets aerosolized into the air and then breathed in by large numbers of people. So since we started air conditioning buildings, the number of cases of Legionnaire's disease and Pontiac fever that are caused by Legionella pneumophila have exponentially increased. Plus, we didn't know what it was prior to that big historic outbreak in 1976, Legionnaire's disease. Another example is, this is another bacterial example, but you guys ever heard of toxic shock syndrome before? Yeah, what is that associated with? Tampon use, yeah, exactly. The fact that most people in this room have heard of it and are like, yep, that's tampon-associated toxic shock syndrome, is evidence that public health education campaigns worked for that problem. It came on the scene as an issue corresponding to when, this is like 40 or 50 years old now, but when corporate with companies that were producing tampons started to formulate these ultra absorbent types of tampons, they put it on the shelves, and then girls were coming down with this potentially fatal septic condition called toxic shock syndrome. It's a toxin-mediated disease caused by Staph aureus. It befuddled epidemiologists who were looking at the types of people who were coming down with toxic shock syndrome. But big investigation, they finally were able to link that medical problem to what was causing the problem, which was these ultra-high absorbency tampons. So they let the companies know, they let the corporations know. The corporations adjusted their products. They started including information about the risks in their pamphlets. And we also started teaching this in just normal public education, public health education. So everybody in this room heard about that somehow, but public health education was a big part of that. And then now, most of those cases that we have aren't even linked to tampons. They're linked to wounds and other focal points of entry for Staph aureus. And then lastly, increased immigration, international travel, increasing globalization. An example of that would be Mycobacterium tuberculosis and imported cases of malaria. Increased travel, I could probably name off another 12 different pathogens that we normally don't see within our borders, but then do get imported into the United States. We get cases of malaria here. I've seen them at, when I was at Madigan, but those are import cases, travel-associated cases. Mycobacterium tuberculosis, we do a really good job here limiting the percentage of the population that has it. So most of our cases are also import cases for Mycobacterium tuberculosis. Holy cow, it's 11:57, guys. I'm so sorry. And you guys have an iPass meeting? Okay. This is a good stopping point. We meet tomorrow at 1300, right, Jake? Yeah. I'll probably just go over this slide and not the rest of them tomorrow, but we do have a review for exam 2 tomorrow. So if you have time tonight, I suggest looking over that material, trying to come up with some questions or some areas that you're weak on. It's kind of a compressed timeline for block 2, but we'll just do the best we can. Okay, thank you guys. Sorry for running over. Yeah, sorry about that. We'll see you tomorrow.